this talk, Dr. Bharti Mangla. Uh, so the title of her talk is Development and In Vivo Biodistribution Imaging of Lipid-based Nanoformulations for the Treatment of Breast Cancer.
A very good afternoon to all. I am Dr. Bharti Mangla. Today the title of the presentation is the Development and In Vivo Biodistribution Imaging of Lipid-based Nanoformulation for the Treatment of Breast Cancer. As we all know that I think a lot of presentation has already going on that every 4 minutes a woman is diagnosed with a breast cancer. So there is a lot of stages of the breast cancer, stage 0, 1, 2, 3, 4, 5. There are a lot of stages of the breast cancer. And still now there are exact reason of the breast cancer is still unknown. So there are various types of breast cancer is also there, non-invasive breast cancer, invasive breast cancer, less common or rare cancers are also available in the breast cancer. So if we talk about the treatments, there are various treatments are available for the breast cancer like local treatment, systemic treatment, common treatment approaches are also there, even a combination therapies is also there. But what is the reason that breast cancer is still a leading cause of death? So there are a lot of challenges in these treatments that it can, if we talk about the chemotherapy, so it has a various side effects of the chemotherapy. If we talk about the radiation therapy, then it's also have a challenges like a skin changes. If we talk about the hormonal therapy challenges, so it has also itself has a challenges. If we talk about the targeted therapy challenges, itself has a challenges also. So what we can do that we can shift it to a nanotherapy. So nanotherapy, they can help us to deliver a drug directly to the site specific, like on the breast cells. So this is the major, we can move shift to the nano through help of a passive and active targeting. Also, if we deliver a drug through the nano, then it reduces the systemic toxicity. Also, it helps in the improve bioavailability. Also, it enhances the drug stability. Or moreover, it can help in the controlled drug release. Personalized medicine we can also do with the help of a nano delivery system. Or moreover, we can overcoming the drug resistance, which can appear with the chemotherapies. So there are various type of lipid-based nanocarrier systems. If we talk about the commercialized based nanolipid carrier system, yes, commercialization is also available. Nanolipid carrier system. So here is the list of the commercialized based nanolipid carrier system which can orally administered for the different type of diseases, not for the breast cancer. If we talk about the breast cancer therapies, yes, still there are 2 to 4 nano formulations which are available in the market. So if we talk about the mechanism, how it acts, so I'll show in the next slides that I have prepared the nanolipid carrier system of the various anti-cancer drugs. So what we do that we did the in vitro characterization, we did the particle size, zeta potential, surface morphology, entrapment efficiency, and we also characterized the formulation with the help of a DSA in vitro release. Here is the results of our research formulation, that what we have found that the formulation which we have developed, it is of negative charge, because the charge is very important that as we can administer in our body. So charge is very important to determine it. And if we talk about the particles that we have make it into a nano formulation, so SAM and TAM is the major study to find out the particle size or its morphology. If we talk about the differential scanning calorimetry, what we can found from this study that either our drug is in a amorphous form or crystalline form. So what we have found that the prepared nanolipid carrier system are converted from the crystalline to the amorphous nature. This is the in vitro release. From this study, we have confirmed that the prepared formulation follows the control release pathway, which is very important when we are preparing the nanolipid carrier system. And if we talk about the modeling, then it can follow the Higuchi model. If we talk about a stability study, this we can store that drug for 3 months to 9 months. So stability study, it is very important to do the stability study, and we have found that our formulation can be stable at all 3 temperatures, which is of the 4 degrees Celsius, 25 degrees Celsius and 40 degrees Celsius. This is all the accelerated stability testings. Then also we have performed the human breast cell line culture study on MCF cell line, which is a breast cancer cell line, and we have found that the developed formulation, if we compare with the plain drug and the nanolipid carrier system, that the cancer cells are more killing through the nanolipid based formulation. And we also do the cellular uptake, that which we have developed the nano formulation, then it can completely intake higher than the plain drug. We also did the preclinical studies. Through the preclinical studies, we have developed the breast cancer on the animal model, and then we have treated that DMBA induced animal model breast cancer through the formulation which we have developed it. So here is the results, and histopathological studies is also showing in the slides. And also we have confirmed that we have taken the nanolipid carrier system through the orally. So oral bioavailability is very important, that it can either enter into a blood or not. If it cannot enter into a blood, so their efficacy, it starts to reduce. So through this study, we have confirmed that our nanolipid carrier system increases the bioavailability as compared to the plain drug. We also did the chronic toxicity studies for the 28 days. We can give the drug daily to the animals for 28 days, and we have confirmed that the that drug can affect the organs like liver and kidney, but if we can incorporate that drug in the nanolipid carrier system, it can not affect the vital organs. We also did the confocal laser scanning microscopy, and we have did the in vivo biodistribution study. This is the main mechanism, that how it enter into a body. That as you show in the figure, that we can take the nanolipid carrier system through the body, then it follow the lymphatic pathway system. Lymphatic pathway system is the intestinal pathway system. So it treats the breast cancer through the lymphatic pathway system. So this is the mechanism, that how the nanolipid carrier system enter into a body, and it cannot affect the organs. As you see in the C group, so this group is our nanolipid carrier system group, which shows that formulation can directly enter into a body, and then it can directly enter into intestine, not follows the organs. So through the reason of interest, we have confirmed that the nanolipid carrier system directly follow the lymphatic pathway system. So this is my conclusion, that lipid-based nanocarrier system, yes, itself, if we talk about the nanolipid carrier system, yes, it can be a treatment, it can prove to be a breast cancer treatment nowadays. So we can go through the nanoshift also for the treatment of breast cancer. So these are my research highlights and my publications, and I have successfully developed the 9 formulations for only the breast cancer, and I have uses the 10 anti-cancer drugs, and we can also performing the topical formulation we are trying to develop for the breast cancer. So thank you so much.